the screen to get it moving again.
Okay.
So I'm delighted to introduce to you CodyBagnall. So Cody got his undergraduate degree here at Texas A&M, but in aerospace engineering. And then he saw the light, and I'm really good. And turned over to the agriculture side, but he stayed in engineering, so he got his master's degree and is currently working on his PhD in bio and ag engineering, and he is expected to graduate really soon.
I hope so.
Yes. May 2020, and he's going to talk to us about super high tech sensors. So using MR magnetic resonance to detect.
Good afternoon, everyone. I'm going to talk about our field-deployable magnetic resonance imaging rhizotron, which we're wanting to be able to use for enhancing root growth and function. The goal of this project is to develop a field-based low-field MRI system that we can use in situ. Now, when I say low-field, if you go to the hospital and you get an MRI done on your knee, they're going to be operating somewhere between 1 to 3, 1 to 5 Tesla, so magnetic field strength. We're operating at 47 millitesla. We dropped the field strength way down, and we'll talk in just a second about why that's so important. So people have been trying to use MRI in agricultural and in soils in particular for a while now. There's some problems with that. Main one being the magnetic material that's present in most soils. The literature kind of says that anything over about 4% of this magnetic material by mass distorts the image, and they're pretty much garbage. You can't see anything of any value. Another study looked at it and quantified it just a little bit differently, which is anything over about 10% high clay content, which means all of our natural soils that we care about growing anything in, it's kind of out the window. Just for a little bit of reference, we're doing this with energy sorghum, which I've got out in the field right now. So the way the MRI works is that we create a stable, homogeneous magnetic field, usually an electromagnet, but you can also do it with a rare earth magnet. And we'll take a target, something that we want to image, and we put it in that magnetic field. And what this does is it aligns particular, in our case, hydrogen nuclei, all in the same direction. And then we can apply an RF pulse, a radio frequency pulse, and we can get the spins to all be in sync. And that's actually what we're measuring. So when you look at an MRI image, you're actually seeing those elements that you've got all lined up. Now, you can also do it with other elements. You can do it with certain types of carbon. C13, I think, is a fairly common one. But over time, called relaxation time, these nuclei go back to their original orientation. And in so doing, they end up canceling each other out. And this is going to become very important later on. So big things to take away here are that we're only looking at hydrogen. So essentially, in our case, water. So that's the only thing that we can see. And we need the thing that we're interested in seeing to have a very different relaxation time from anything else that's present. In our case, that's soil water. The other thing that we're interested in is what size of the things we want to see. In this case, we're looking at roots. So John Mullet's group has been doing some work with this, which is where these graphs come from. And what we know is that the nodal roots are 1.5 to 2 millimeters in diameter on average. But as you move away from the plant and the soil surface, these roots get very small very quickly, down to about 0.3 millimeters in diameter. So that's kind of the range that we would love to be able to image and collect data of. So where we started is by looking first at relaxation times, and I'll show you that data in just a second. And then the other thing we did is we created sort of a benchtop MRI, low field MRI, which are the images that you see there. This one's got an 8-centimeter board. It's about 30 centimeters tall. You can see the electromagnet, and then the other things there are gradient coils, which are used for orienting where things are in space. And then you also see the RF coils there. And what we ended up doing is we took some cores and shipped them off to New Mexico, where our collaborators were building this thing. and we had them run some images and see, can we get around this magnetic material in the soil problem. Now you'll notice that I've got some percent clays there. The reason this is important is because literature says anything over 10% is no good. So these are soils that either came out of the field that I was working in, or in one case, the Houston black came from some rhizotrons we planted from Sorghum. So relaxation times. To measure this, we took 6 different soil types at a variety of clay contents, and we're running them up to 7 different water contents. And what you see there is that the relaxation time, T2, which is on the y-axis, is different for each soil type. And it's got a linear relationship with water content. But the nice thing is that no matter what the soil was, the relaxation time is much, much smaller than that of roots. So if you look up there in the top right corner, the T2 soil max is just a little over 4 milliseconds. That's the longest relaxation time we found in all the soils that we looked at. The T2 roots is 85 milliseconds. That is the shortest, the fastest relaxation time we found in the roots that we looked at. So what this means is, practically speaking, when we go to image, we hit the sample with our RF pulse, and then we wait, say, 7 milliseconds. In 7 milliseconds, all of my water has relaxed and is now canceling itself out, allowing me to now start collecting data at, say, 8 milliseconds. From 8 milliseconds to about 120 milliseconds, I'm collecting just root signal, which means we can distinguish between soil water and root water. So this is a good start. The next stage then was to see, can we get around the clay problem? And it turns out we can. So these are cores taken from 0 to 70.5 centimeter depth, either side of the plant between rows. So these are mirror roots, which means they're approximately 2 millimeters in diameter. Resolutions, 0.6 millimeters. These scans took an hour to create. So just sit there collecting data for an hour. But what this shows is that the low field does get us around the magnetic susceptibility problem. And part of that is because of the low magnetic field. The problem is that it also means low signal strength. So our signal to noise ratio is much, much lower than, say, medical MRI. We kind of get around this by long scanning times. Nobody wants to sit and get their knee MRI for an hour. The roots don't seem to care. So we can get images. Now the next question is, can we get it in the field? We did take some more images in some other types of soils. This is a Houston black and a manufactured soil that came from Aggieland turf. These are a lot faster, 15-minute scans. The other thing that I'll go ahead and talk about the data for a second. These are called 2D projection images, which essentially means that the information on the back side of the image is present and can be seen even in the foreground, which makes things really interesting, and I'll show you some more pictures in a minute where it allows you to get a lot more information in one image if you sort of know how to deal with it. These are also a coarser resolution, which sort of helps with the decrease in scanning time. So the other thing we had to do is we had to figure out what the balance is between how big our pixels are, how long it takes to scan, and exactly what our signal to noise ratio is. So if you ignore the bottom row and just focus on the top row of images, as you move from left to right, we go from 1.6 millimeters to 1.25 to 0.6. So we keep our scanning time constant at 32 minutes. And as you go from left to right, the image gets grainier or snowier. That's our signal to noise ratio getting worse. If you drop down to the lower row of images, we'll keep the same step size of pixel resolution, but we keep a constant signal to noise ratio across all 3 images. And what happens is we go from half an hour to 4 hour scanning time. So we've got to balance these 3 things, probably dependent mostly on exactly what you want. What size roots you're looking at, those sorts of things. But that is important when you start looking at actually collecting data that you want to get root metrics out of. So we know we can do it at small scale. Can we do it at the big scale in the field? So we fabricated a full-size magnet. This is one that I've actually got in the field right now. It's got a 25 centimeter bore, just a little under 4 feet tall. And we created 2D projection images of our 2 field soils. So I collected 6-inch cores, 6 inches in diameter, 6 inches tall, shipped them to New Mexico where they imaged them when they were calibrating the system. And so each rotating image is a collection of 8 2D projections, which gets you 180 degrees. And then by inverting the pixels through programming, we can visualize the back 180. That's what creates this sort of rotating frame. These 2D images take 2 hours to get a rotating image like this, 2 hours and 10 minutes. Compare that to true 3D data, which takes about 9 hours to collect. So depending on exactly what you need, if you want, say, just branching angle or root length or total biomass, you can get all of that much quicker using this method than true 3D data. So now we know that the big magnet works. We know that we can take the cores into the lab where there's no noise. We can get images. Can we do it in the field? That's the next question. To do it in the field, what we decided to do, you pick a plant that you want to image, you chop it off at the ground level. You take this green PVC pipe that is about 36 inches tall, and you center it over the plant, and you push that into the ground with this Giddings probe and the homemade cap. And then you dig out an annular-shaped hole. That's what you drop the magnet down in. And if it's nice and wet, you can use this core. And if it's our typical dry summers, you do it by hand. And it takes a while in the play. But we dig to about 24 to 26 inches, and then we'll image the 10-inch depth. So the MRI is imaging in the center third of the magnet. So you have 2/3 down the ground and about 1/3 above the ground. The next thing you do is you pick it up and you put it in the hole. When it's got all the coolant in it, it weighs about 1,000 pounds. And so we built a lift system that I can center the magnet within a 1/16 of an inch anywhere inside of a 3 foot by 3 foot area. So I have a tractor with forks on it. I get close, we get out, and by pulling on some chains, you can have it walk exactly where you need to go. And then the next thing you do is you hook it up. And so if you look at that bottom right-hand picture that's hooked up and ready to go and about to start imaging, we have a nice little trailer out there. We're running off of 2 generators. Each one's about 12.5 kilowatt. And that's more than enough to run everything that we've got going out there. A couple of power supplies, 3 amplifiers for the gradients, coils, and then 1 amplifier for the RF coil. These are essentially just the same amps that get used like if you go to a really nice concert. It's just been used for this. So once we get in the field, we can start making images. Now these images are technically above ground. So we set the magnet on top of the ground out there, run the generators, pulled some 10-inch cores, so they're 10 inches deep, still in the green PVC, push them in, dig them up, set them in there, and you can get these very nice 2D projection images, both in the Westwood loam and the belt clay. So the next step is to actually do it in ground around a column of soil. Now the problem with this is that there seems to be a bit of environmental electromagnetic noise. Not entirely sure where it comes from. My money is on the pumping jacks that are out there, like 200 yards from the hole, but we don't know for sure. But what we have figured out how to do is the noise is transient, meaning that it doesn't end up in the same place on the graph each time. So if you look at that top right-hand graph, it's got a bunch of noise in it. And so we take the same line of data 8 times. We have 8 replications. And because the noise is transient, when we compare those, we can actually mathematically pull the noise out. And you end up with a graph much like this one. Now, there's 16 spikes on here. Those are our 16 echoes from the echo train. And essentially what you're seeing is you're seeing the decay as those hydrogens go back to their original spin state. That's why it's disappearing. After those 16, we'll hit it again with an RF pulse. It'll pop back to here, and you can do that. That's how you do your signal hydrogen. So this is kind of the point that we're at. I actually was out in the field today collecting in-ground data, and we're working on processing it and seeing just how good it is, how much error is still there. But essentially what we know at this point is we know the size of the roots we want to get. The small is 0.3 millimeters. We can easily see the nodal roots, anything from about 0.5 millimeter, 1 millimeter, are very clear. We need to be able to balance the signal to noise ratio, the resolution, and the scanning time. This will sort of determine exactly how small we can detect. We know that we can collect images in well over 10% clay. In fact, we've collected images in everything we've tried thus far. We have found some noise, particularly when we're out in the field and in the ground, but we seem to have found a pretty decent way of post-processing the data so that the information is still good. And we know that we can collect it both just sitting on the ground, which is quite a bit faster in terms of just pulling it up and keeping a lot of samples going. But we can also do it in ground around a column of soil. The next thing that we want to do is actually start pulling information out of these images. One of those is biomass estimations. We do this in two different ways. One of them is called 0D imaging, which is essentially no spatial information at all. It's just there or it's not. The other one is called 1D imaging, where we're looking at spatial information but just in the vertical direction. So now we should be able to estimate how many grams of roots are in the top 3 centimeters, top 5 centimeters, whatever it is that you want to look at. And then, of course, things like root length, root diameter, things like that. And then the other one that I want to mention, though I'm not working on it, I'm sort of providing some basic data and imaging, is the application of a neural networking software to allow us to do undersampling. And by undersampling, if we can undersample and get as good of information, then we can either scan faster for the same quality of data, same signal-to-noise ratio, or we can improve our signal-to-noise ratio and keep the same scanning time. So we can double the signal-to-noise ratio and still scan in 15 minutes, which should improve things like how fine of a resolution can you actually get. So that's something that our collaborators at Harvard are working on. And with that, I'd just like to leave this slide up, which is the team that is currently working on this project. There's been a few iterations as people come and go and those sorts of things. And then recognize our funding agency, which is the Department of Energy ARPA-E Roots project. And with that, I will end and answer any questions. Yes, sir.
So, I realize this is to explore the concept, but at some point, you envision getting this to where it would actually be easier to dig roots out the western. What you're showing me right now.
Yes. Yeah, I mean that's sort of the goal. Right now it's ground truthing and knowing what our error is. But the goal is that you would just image, know that you have plus or minus some error in root length and biomass. And then the other thing is that this portion of the project, hopefully, is a stepping stone to a one-sided magnet. So we wouldn't do any digging into the ground at all. Now that all depends on a plus up and funding agencies and politics beyond my pay grade, but that's sort of the goal, is to not have to do any digging whatsoever. Yes, sir.
How safe is this to operate? You're near a big magnet for a long time, so does it have any effect on you? That's one question. Then the second question is, will you ever be able to do this non-destructively? Because we want to see if we can filter out the biomass, above ground biomass, and just be able to estimate the root parameters.
Yeah. So to the safety issue, this one, and this is actually one of the really nice things about low field, is this one's quite safe. Both in terms of just it has no real health, no known health problems being around it long term. But the other thing is I accidentally stuck a screwdriver down in there, thinking the magnet was still on, and you could feel it. It's kind of weird to be able to feel the magnetic lines and feel the different ways that it pulls, but it by no means pulled it out of my hand.
You have to be method free when you're operating, like take off your watch.
No, see, that's the nice thing is it's low enough. This one is a low enough field. Now others, yes, you do, but this one's low enough field that, like I stuck my hand in the bore with a screwdriver and had no trouble holding it, and I was actually using the screwdriver to pull something else out. Yeah, so it's very, very low magnetic field. But then to answer your other question about non-destructive, yes, you just have to be a little more thoughtful about how you do it. So one of the things that I know Dr. Morgan and Dr. Mullet would like to look at is long-term growth changes of the plant as it grows, where does roots go, how does soil structure impact that, clay pans, things like that. To do that, what you'd have to do is you have to plant the seed in that spot, or put your core in pretty early, and then come back and image over time. The other thing would be the one-sided magnet that you would just have on top of the surface next to the plant. You could also do non-destructive. Yes, sir.
You talked about software that speaks like the blood How fast is that?
That's a good question that I don't really know the answer to, because it's not only a function of the auto map, which is the programming system, but it's also a function of a couple of other parameters. But if you just straight compare them, the numbers that I've heard is half again faster, but that is sort of a rough estimation from what I can see.
So the goal is to make it probably easier to faster.
Yeah, so I think there's a little bit of a balance there between, do you want it faster, or do you want it finer resolution? Do you want it, if you want sub-millimeter resolution, and if you want to be able to very finely map the architecture, are you willing to wait an extra hour to get that information on the single plan? If you want to go more towards a high throughput phenotyping type methodology, you definitely want it faster, but you'll probably be sacrificing some of the information that you might otherwise be gathering. So like most tools, sort of a trade-off between what you want. Yes, sir.
Have you tried adjusting, say, your magnetic field strength and seeing what difference that would make?
Not in this model. So this is a straight room temperature electromagnet. The RF coil is tuned to a particular frequency, and to change that, to change the magnetic field means to change the tuning, which means pulling the actual RF coil apart and redoing it. We are in the process of getting a superconducting magnet, which will allow us to change magnetic field over a fairly broad range. So technically, low field is anything less than 1, but we'll be able to go almost all the way up to 1 and down a little bit below where we are now.
Yeah. Have you thought about using other isotopes? Or do you think about other?
It has cropped up on many occasions. So people have thought about it, yes, and it's one of the things that's gotten batted around in discussion of how could this be used, where would it be used. I know Dr. Mullet has mentioned a number of times of being interested in using other isotopes to help see things more clearly and to look at very specific things.
Yeah, I would imagine combining the hydrogen with this project would get much more redundant.
Yeah, I was actually talking to one of his graduate students who talked about it. You can actually, there's a process which you wouldn't want to do in the field at all, but you could apply a heavy metal, which then binds to the roots, and then you can see it better. Again, it all depends on what you want out of it. Yes, sir.
But also, what the field of water comes in, sometimes the measurement of
Effective measurements, well, impact and measurement, yes. In that, we have to change our imaging sequence a little bit. So the relaxation time of soil water is much shorter. But if, say, I start scanning at 4.5 milliseconds, there is a little bit of residual signal as it's dying out, which shows up as noise in our image of roots. So you do have to be aware of what that's going to be, which is why we make set it at like 8 milliseconds, so almost double what the relaxation time actually is for the soil water, just to be sure that we get that all filtered out before we start collecting our root data. And that's helped by the fact that our root data is so long. If it were much narrower, we'd have to come up with some more creative methods, which you can do. I mean, that's one of the things that they run into when they do lung MRI. The relaxation times are all very close between lung, muscle tissue, skin.
What's the reason why water in fruits have a longer relaxation time than in soil?
That is a good question that requires quite a bit more research. There's been a couple of hypotheses about that, but I don't have an answer. As a layman engineer on the project and not an NMR physicist, I really don't know. I know that there's a theory that the water is reacting with the soil surface, and in particular the paramagnetic material or paramagnetic material in the soil, and that is what is creating such a short relaxation time, and it is why across the board the relaxation time increases as water content increases, because the new water sort of added to the system is not in contact with the soil surface. But again, straight theory, no testing has been done on that theory.
You mentioned that you just arrived with magnetic field. So most of the people work on only talking about the microfields.
Yes.
What's your frequency?
We are at 2 megahertz. Well, so most of our stuff is at 2 megahertz. We also did the soil water was actually done at 11 megahertz, which is 0.2 Tesla. Yeah, 0.2 something. Yes, sir.
Where do you plan to publish this work?
The first manuscript, I hope, goes out in the next week or two, in terms of going to the journal, and that will go to Geoderma. Then we'll have 2 more that'll be focused on the field aspect. I don't know where that's going.
Oh, I have some suggestions.
Okay. I'd love to hear.
Okay. Plant Genome Journal would be great for this.
Okay.
Really innovative work.
I think the question for thinking this art purpose would only to map soil.
Yeah, I know Dr. Morgan would love to see it get applied to mapping soil water. That gets a little bit more complicated because you get into really, really short relaxation times. But one of the guys at 82MR, Dean, has focused most of his work on short relaxation times. So, yes, sir.
I just wanted to make sure I heard you right about the power requirement. You're using 212 kilowatts.
That is correct.
24 kilowatts.
Yeah.
And were you really using all that power, or is that?
No. So the original plan was to use basically 19 kilowatts, 18.5 kilowatts. We added in a chiller to keep the magnet cooler, because we're generating about 2,000 watts of heat energy. So it's not just to be safe and not turn my $100,000 magnet into a paperweight. Let's put a chiller on it. But with that, we had to change generators, and if you're going to change generators, the extra 6 kilowatts was the same price. So we do have a hefty abundance of power.
So you're closer to 20.
Yeah. Probably, 20 is probably pretty close. Yes, sir.
So when you image like in the inside along the surface, you saw all this noise that came in.
Yes.
Any kind of dirty tricks that you could try to apply to get rid of that, like the outer spot, the big hole covering the inside with a little spot or something?
Yeah, so we went round and round about that, and basically what we've come to is that there's no good way to filter the noise before you collect the data. The reason is because we believe, and we haven't done extensive testing on this, but we believe that essentially what's happening is the noise is traveling through the ground. You've essentially, because you have a column of soil that's connected, you've essentially got an antenna sticking up in your RF coil and gradient coils where you're collecting your signal. So you're basically piping in noise with that. So, I mean, if you cut the column off and pull the whole thing out, or even just cut the column off and put shield down, you would not have that noise. Which is why you can see the sort of above ground images that I did don't have any noise. Because that's what we did. We just pull it out of the ground, and then you don't have that antenna, so to speak, sticking up inside of the core. So the best that we've got thus far is to pull the noise out in post-processing. And like I said, the way we're doing that is basically by taking 8 duplicates, comparing them, and then using that to mathematically essentially chop off the noise spikes and not have it in the data. So that's sort of our method. That does take longer because we're not allowed to average as we go, which sort of speeds up our scanning process. But it's not significant. I think right now images take 16 minutes instead of 15 minutes. So not a huge addition. Yes,
After you take the image, you collect the core and wash it and look at the parameters and then correlate.
yes, that's
And
what
then you also add GPR and see if it compares to this.
So the current idea is, yes, so we take the 0D, the 1D, and the 3D images, and then we pull the core, and I've got an undergrad who chops it off at the imaging zone, the 10 inches, and we wash it. And we're actually sending these through, Dr. Mullet has a WinRhizo scanner, and so we're sending it through there, and then we're doing wet weight, dry weight, so we can correlate, and that's sort of our ground truth. What does WinRhizo say we have in terms of root diameter, root length, these sorts of things? What do our scales say in terms of dry mass, wet mass? How can we build a calibration equation and use that to create good data? And how do we know what our error is? That's it. Yes, ma'am.
So when you're looking at, so the hydrogen thing was really good.
Yes, ma'am.
So with that being said, if you're looking at different pH, soil pHs, maybe help influence some of your imaging, or maybe your plant who's affecting the rhizosphere by hydrogen helping out to acidify the rhizosphere around itself. So maybe looking at different plants who maybe have a better manipulation of their rhizosphere by acidification, maybe helping imaging.
It's possible, yes. At this point, we've actually imaged in 4 soil types, all mostly locally. 2 out at the plant center, out at the farm. 1, Dr. Morgan pilfered some Houston black clay from somewhere, and then the other being a manufactured.
Did you see any correlation in terms of the type of soil that may be better for imaging?
So, in our stuff, no. In the literature, the less clay, the more power you can use, the better the image is. I don't know that anybody's really dug into the soil chemistry. Most of the people who have been doing this work have been NMR physicists. There's a few groups, most of them are out of Germany, who right now I've seen some recent publications, 2018, 2019, looking at high field 3, 4, 5 Tesla. Most of those are manufactured soil stuff. So greenhouse grown, potting mix of some kind. But to my knowledge, I haven't seen anybody really look at the effects of soil chemistry or plant chemistry, as the case may be. We've only tried imaging 1 plant type. We've only done energy soil at this point.
we need